This topic that I will be speaking on today has been pretty much my life work, and it's evolved quite a bit. And as fast as farmers have evolved methods, we in soil labs are having to evolve our methods. And it's a whole movement that's going on in the background. If you're not aware of it, it's caused quite a bit of discussion and debate. There's a lot of challenges. If you ever felt pushback on new methods and innovations that you were trying to make on the farm, we do the same thing in the world of science and soil labs as the people who want to stick to the old methods and don't understand the need for new methods. And it's quite an exciting arena for discussion right now, as Ray and others this morning so well highlighted. So just to go down memory lane here, I want to pay tribute to St. Louis today for a study and an event that changed my career a long time ago and really shook the American agricultural establishment to its roots. And it was this study, if you can see, it was published in July of 1975, right here at Washington University, by a group called the Center for the Biology of Natural Systems. It no longer exists here. It got moved back east. It was funded by a special dispensation of Congress. They were going to set up 6 of these centers around the country, and they only ended up funding Washington University to do this. And they published a study. I'll just read the title because it galvanized a discussion that is going on to today. A comparison of the production, economic returns, and energy intensiveness of Corn Belt farms that do and do not use inorganic fertilizers and pesticides. The brainchild of Willie Lockeritz here on the right and Barry Commoner, who died about 5 years ago. What this study did was they paired farms here in the Corn Belt and looked at how much energy is going into producing the crops. And the stunning results are that caused so much controversy then. And I see a lot of you in the audience. How many of you remember this study? Does anybody? A couple people? I got in my car and drove all the way out to St. Louis to meet Barry and Willie Lockeritz, and I've kept up with him over the years. And I said, I want to go to school here, and this is the kind of study I want to do. And they said, well, you can't exactly do that, but talk to Albert Schatz here. He has a new university program for things like this, and they sent me to Europe after this. But what they showed was this intensive use of nitrogen fertilizers and lots of tillage made the inorganic farms, the conventional farms, 3 times more energy intensive than their paired organic farms. Now, I'm not here today to start a discussion about organic and non-organic farming, but what I am saying is that I am seeing innovations that you all are using with no-till and cover crops that probably, if we did a study like this again on energy intensiveness, we would show that you are evolving a method of farming that's significantly more energy efficient. And this study, which earned so much criticism in the country, it's almost hard to imagine why there was so much severe criticism. They went on to publish it in all the major magazines. And Willie Lockeritz once shared with me the critiques and the letters he got and the condemnation of this. And so if you've ever felt pushback in any innovation you're doing, well, people before you have had the same experience. And this study really helped grow all these new movements. It didn't just provide new energy for farmers who chose to be organic, but I believe it's at the core of what we are trying to do now, particularly as we increase soil health. So, to the main question here, what makes your crop grow and what makes your soil fertile? You're all familiar with soil testing. I've been doing it. I started soil testing when I was 18 years old. There was something about it that I just loved, and I started my first laboratory as an undergraduate in agronomy, even though they told me I couldn't do that. And I started testing soil, and I was fascinated with the concept of soil biology a long time ago, and the customary preoccupation with a simple nutrient picture as the primary mechanism of managing soils and crop growth. And most labs then and still today do not test nitrogen. They just tell you how much nitrogen to put on to replace the nitrogen your crop will remove. And Commoner and Lockeritz's study faulted the high use of nitrogen in 1975 for the reason that modern farming was so energy inefficient. Now, since 75, nitrogen rates have continued to climb, as have yields, but it could be a zero-sum gain. If we did these studies again, what we would show is that the efficiencies are coming from soil health, from things that you're doing to reduce the amount of work time in the fields, and I hear a lot of farmers saying they're saving so much time with no-till, but I'm also noticing they're using less fertilizers as the soils get better. That's the efficiency cycle. So here's my problem. Modern emphasis is primarily on chemical factors for growth. The rest is taken for granted, right? I mean, couldn't you put it that simply? And if you do, and if you look at the reports, and I do this too, it's just minerals, minerals, minerals. We're always talking about minerals and what we need to grow our crops, and it all comes down to the inorganic nutrients that crops require. Why is it that we're so silent about soil health? Why is it that the whole microbial underworld is not being discussed or is considered, as this suggests, to be taken for granted? So here's a picture of what I call the soil biology underground. And this soil biology underground is the source of everything that I will lump together today called soil respiration, the breathing of the living soil. And if you break it down into two quadrants, you have the content of organic matter in your soil, which is largely comprised of plant roots and litter in the process of decay, soil organisms, 5%, and humus, which is decayed litter that has been transformed by microorganisms into a poorly defined, high molecular weight structure that binds soil particles and has longevity up to 1,000 years in soil. But only 5% of that is microbes. So that may sound like a very small amount, but if you break the microbes out and look at what's going on there, and this is just an approximation. This was from a German soil biology institute that did this and gave it to me recently. They broke it down into algae and fungi in one group, bacteria, actinomycetes in another, and soil animals. And you can see that the soil animals portion is significantly less, of course, and that varies and depends on your management practices. Certainly, there's much less soil animals if you're in a tillage system, and the thing that's really impressing me as I study no-till farms is the increase in the earthworms that we're observing. So the animal content is changing in the soil, but we see very similar ratios of bacteria and fungi. But so there's this whole world in the soil that we've been ignoring for so long. So here's an attempt to quantify it, and I owe some of these numbers here to Jim Horman and Rafiq Islam at Ohio State University. They put this out in some literature on their website a little while ago. When you hear people talk about the numbers of microbes in soil, it's difficult to comprehend. We say 10 to the 8 to 10 to the 9. 10 to the 9 is a billion organisms. 10 to the 5, 10 to the 6 fungi, that's 100,000 to 1 million. These numbers are huge, right? And it's hard to relate to them. And you often hear people say in 1 teaspoon of soil is more microbes than human beings on the whole planet. And I ask people what that says to them, and they say, we don't know how to relate to that. That's a dimensionless number. So it's more interesting to look at the biomass. The biomass is what would they actually weigh if you could measure them? And I took their biomass figures and then I converted it into pounds an acre. And again, this is just I took the ranges that they have and I compared it to some tests that I had done recently that modified them up and down. And so you could have a little more than a ton of bacteria, a ton of actinomycetes, nearly a ton of fungi in a really healthy soil, and much less just in pounds of algae, protozoa, and nematodes. But add that all up, that's worth several animals per acre. Now, there's a very popular comic going around the country on how many cows are hiding under your soil. And one of the things I just threw this cartoon up here is, this is their niche under the soil, but the size of these guys is 1 micron. That's like 4/100,000ths of an inch. That's why this is so difficult. We can't see these organisms. If this were cows, there'd be 1, 2, 3, 4, maybe 4 or 5 cows per acre. You'd see it, you'd feed it. But what you don't see ain't there. And this whole thing about these hidden organisms and microbes in the soils, what's so challenging to me is to try to make this more visible. And once we make it visible, to develop the methods of quantifying it meaningfully, to get reproducibility and quantification, and then start relating it to your practices. And what we are trying to do with soil labs around the country is change the methods that they're using in the labs. Now, you heard some discussion this morning by Ray about carbon sequestration, and this is a huge and significant discussion. Carbon is really the niche of microbes, although there are microbes that don't feed on carbon in the conventional sense, but they transform inorganic elements. If you look at the Earth, soil is the largest active reservoir of the world's carbon. It is a very, very significant fact. This is billions of tons of carbon, also called gigatons in climate carbon discussion. 1,580 billion tons of carbon. It could be, I've done the numbers by several different studies, anywhere from 1,500 to 2,500 gigatons of carbon are sitting in the topsoil around the planet. And if you add up the entire atmosphere, there's only 750 or half of it. So if, just hypothetically, if we killed our soil tomorrow, just killed all the organisms and let all that carbon go into the atmosphere, CO2, we would have only months to live. It would triple the CO2 in the atmosphere that quickly, and we would barely be able to survive. That's how significant soil is. It was this extraordinary repository. Then you have all the land organisms which have as much carbon in them as the whole atmosphere does. And then you have the warm oceans, and I didn't include the deep ocean because there's a lot of carbon there, but it never comes into circulation. But here's the fun part. Who's doing all the respiring? Who's doing all the CO2 respiring? And I had to search through a number of databases and do some calculations here. But if for a minute you ignore insects, and I'll come to insects, microbes, this blue wedge here, are doing the lion's share of respiration. Animals and humans together are only about 1/50th to 1/100th the respiration worldwide of soil microbes. The stunning fact is insects have us all beat. And this I didn't realize until I started adding it up. Insects are doing the majority of the Earth's respiring of carbon, and that's why they're so important. We barely even think of this, the significance of this. 2,140 billion tons of CO2 are being respired. That means eaten and given out every year by insects, and microbes are doing about 100 billion tons of CO2 respiration a year. Now, once again, as long as it's in cycle, it's in balance. It goes into an organism, it gets breathed out. But this is a tremendously significant fact here that makes you appreciate more and more the fundamental role that soil microbes play in the maintenance and the cycling of carbon. So to stand back and kind of create a special picture of this, you could describe the Earth's surface as a living, breathing mantle. And I like to draw these pictures to try help us visualize the whole cycle. And then I listed here a number of the features of this cycle that are so significant and why it's so important to start measuring and appreciating this more. First of all, the soil is food for soil animals. The carbon in the soil is food for soil animals. Secondly, this system is both the sink and the source of carbon dioxide. A sink means it goes there and stays. A source means it comes out again. Most of it is staying in the soil. As we saw from that earlier slide, about 2,000 gigatons of carbon are present as humus in soil, and we really need to keep it there and grow it. It supplies plant needs for CO2, and this is a very startling fact that I have been talking about now for 2 years. The amount of CO2 that comes out of soil directly is affiliated with plants' absorption of CO2 for photosynthesis. And more studies are showing the cycle is very tight. And I'm going to give you some figures on this that makes you appreciate how important soil health is, because it's feeding plants in other ways than just nutrients. Obviously, nutrients, the available nitrogen and phosphorus, are a big cycle here. We talk a lot about the nitrogen cycle coming out of litter. As litter decays, the nitrogen gets released. We thank the microbes for that. The plants get it. But there's a whole phosphorus cycle as well that can be just as significant. If you measure organic phosphorus in soil, it can be anywhere from 500 to 2,000 pounds per acre in organic phosphorus form. So as that organic matter turns over, that phosphorus is set in motion itself, and your plants are going to see it. And again, soil labs don't measure that. We consider it background, and we're not paying any attention to it. Finally, all this cycling of CO2 does something very important. Guess what? It dissolves soil minerals. Carbon dioxide dissolves in water to form a weak acid, and that acid etches the stones, and there's microbiologists that are studying this to see what is the rate that it's etching stone. What's it doing by etching it? It's releasing the minerals that are present in them and setting them into motion so that the plants can get them. How much minerals are present in the geological structure of the soil? The numbers are so vast, you almost can't believe it. There's like 20,000 pounds an acre of potassium tied up in mineral structure in certain types of soils. And the microbes might be getting 10 and 20 of it every year out, but somebody did an estimate on how many years of farming would that supply. Well, you can stretch it out to thousands of years at the kinds of levels that are present. So it starts shaking your picture of sustainable nutrient management when you realize nature has actually got a system in place to feed plants, and we think we're the only ones feeding them. We have to learn to appreciate the dimensions of this. So let's look at this picture. I draw these cubes, and the cube is the atmosphere sitting over the acre of crops. Well, guess what? Microbial carbon dioxide is actually your primary nutrient. All the CO2 that's in the atmosphere, by the way, is microbial CO2, unless it's fossil CO2. And fossil CO2 still accounts for only 4 or 5% of the CO2 that's in the atmosphere. All of it is really microbial respiration, as you saw. Insect respiration, microbe respiration, and human and animal respiration. But look at the rates of absorption of crops of CO2. And this is per day. I have some qualifiers here. Wheat absorbs up to 110 pounds of CO2 per day per acre. And again, my qualifier is I took the 60 most active days of growth for wheat and corn to do these numbers. If you spread it out over a larger season, the number would go down per day. It looks like a bell-shaped curve in reality if you draw it. Corn is even more amazing. 400 pounds of CO2 per day per acre. Isn't that amazing? And that's where your dry matter accumulation is coming from. Now, I have shaken some people by pointing out that theoretically, or actually potentially, all that CO2 is coming from your soil respiration. People have started challenging me, saying, prove it. And I say, well, can you disprove it? And some studies have been done to try to disprove it. And one study in Iowa I found that was done about 10 years ago found that 20% of the carbon in a corn plant, they believe, came directly from soil microbes, meaning the respiration cycle was so tight that the microbes released the CO2 and the corn had it immediately because it's coming up into the lower canopy where the plants need it, where the photosynthesis is so active. And an early study that I found when I was studying in Sweden, they actually quantified this in the 1920s because this was before fertilizers were readily available. And they said, we have to understand this carbon cycle. And they decided CO2 was the most important nutrient, and they mapped it all out. And I've written some popular articles on that. And you can go on my website and get some of that material. But they pointed out that under certain conditions, the soil could be starved of CO2 so that plants are not getting enough CO2 for active photosynthesis. The plants have to pull it down from the atmosphere in order to get it into the photosynthetic cycle if there's no CO2 respiration in the soil. And you can imagine what a dynamic force this is, because wind blows the CO2 across the land, so it's delivering it to the plants from elsewhere. But on a static, still day with no wind movement, the plants are very, very dependent on the close to the ground CO2 respiration cycle. So I think we're going to see a lot more interest in this, because we're seeing differences of a factor of 10 to 1 of a healthy soil versus a less healthy soil in terms of the amount of CO2 that's actively coming out of the soil. And it's not getting out of the canopy. A study was recently done in the tropics that says none of the CO2 being respired from the soil ever leaves the canopy. It's all being reabsorbed by the dense plant layer before it even gets up to the atmosphere. And that's just an amazing fact. And it just shows you how tightly nature has designed this respiration system. So let's quantify it. And I'm going to give you all kinds of examples of quantification here. If we had 50,000 pounds per acre of organic matter, which is just 2.5% organic matter, you could easily have a ton of microbes living on that. And think of them like the cows eating every day. And they are cycling the organic matter, and they're pumping out, using this formula here, about 100 pounds a day of CO2 into the air. If you could wear infrared goggles and walk around your farm, you would be amazed that the CO2 is just streaming out of the soil constantly. And the more alive the soil is, the more is coming out of the soil. At the same time, for each unit of CO2 that's coming out of the soil, the nitrogen that was associated with it as an organic molecule has now been set free. And on a very active day, we've measured the transfers as much as 1 to 5 pounds of nitrogen in one day came out of the CO2 respiration cycle. Now you think of that and how you ignore it when you put down your nitrogen. You think nature is trying to feed your plant, and on a warm, moist day with active soil, 5 pounds of nitrogen could be set free for the plant. That's why this is so significant. And that's why farmers that are getting their soils better and better are telling me we're able to reduce our fertilizer and we're not seeing any reduction in yield. In fact, almost the opposite. And I'll show you one profile on Ray Steyer's farm as evidence of that. The other thing that's so surprising about these numbers, and you can get any young kid who's good at math and say, confirm all these numbers, because it's what we call stoichiometric relationships. Soil's releasing 100 pounds a day, and on average, at full growth, plants need 100 pounds a day of CO2. Isn't that convenient? I mean, this is the amazing thing about the close cycle of soil respiration and plant growth. And I believe that nature designed soil to be the source of CO2 for plant photosynthesis. And what little we have in the atmosphere is just because it's a slightly leaky system. That CO2 got away from the plants. It got blown away, or we left all the worst thing to imagine is bare soils now, bare soils. A bare soil is respiring, unless it's frozen solid, it's respiring CO2, and there's nothing there to use it. There's nobody there to eat it. There's no plant to eat it, so it's going to be dumped right into the atmosphere. Hence all the concern with that, particularly Ray's slide this morning, let's light up these states and make them green from outer space. I'm always shocked flying across this country at all the bare soil I see on farms. It really, you begin to feel it when you think those are just dumping CO2, and there's no meaningful plant cycle there to catch it, to use the nutrients, and to keep the carbon back into the system. How do we quantify this? There's a number of ways to do it. You've heard about the work I've done with Rick Haney, and many others are doing these studies now. you can quantify the nitrogen released from soil by using control plots that have no nitrogen fertilizer and measuring the uptake of N by the corn plants. And here we did a study using CO2 respiration, using the test kit that our laboratory has developed called Solvita, and we represented as parts per million. So if you multiply these by 2, it's approximate pounds an acre per day in the soil. And we measured the corn yield and converted it to nitrogen using 1 bushel equals a pound of N. And look at this close syncrasy of corn yield and the respiration in the soil. This is the basic cycle we're talking about, where the soil is capable of feeding the plant nitrogen. Now, there's a lot of ways that this doesn't work. If the soil is cold and too wet, it's not going to work. If you're not feeding the soil and the respiration is low, you might as well ignore it. I get soil tests in the lab and I say, I'm sorry, your respiration and your organic matter are so low, you need all the fertilizer you can get. You have to carefully manage your fertilizer because I can't guarantee you that the soil will get it to you. But as we push our soils higher, we're seeing control yields come up without the synonymous input of fertilizer. This is one way to quantify it indirectly, and this is where most of the soil scientists and the agronomists are debating right now, is how good is this so-called calibration, and we know it varies quite a lot from soil type to soil type. Another way to quantify it is with your own eyes, and I think this is a wonderful way to do it, to look at this interrelationship. Look at these spurs of dark material going down into the soil. You all know what that is, right? Those are earthworm canals, and they've carried the humus, or they've made humus as they carried the organic matter down, and the plant roots said, oh, we'll take advantage of a good thing here, and we'll follow the channel down. And there are some scientists now just working on analyzing the composition of these channels versus the soil right next to it. There's an amazing difference in fertility. So the earthworms are directly increasing the fertility 4- and 5-fold over the surrounding soil. Plants are taking advantage of it. It's hard to measure this. One of the projects I did as a grad student was I had to count earthworms in soil, and we would cut soil down to different depths and shear off a surface, and you count all the earthworm canals. And this is one study I did, counting the earthworm canals. That's the y-axis, which is the upright vertical axis here on the left, how many canals there were per square meter. Now, you could say a square meter is about a square yard. There's a lot of canals. So here's soils that had 100 canals per square yard looking straight down at the soil. And then we did respiration. Now, I did this study a long time ago, and it was published first in Swedish, and then I translated it. But the numbers of respiration was just a classical titration respiration test, either done in 1 day, which is the red dots, or a 7-day test, which was more common 30 years ago. Everybody thought you had to do 7-day respiration. We now know it gives you the same picture as 1 day or 3-day. And look at the sharp relationship of CO2 respiration with the amount of earthworms in the soil. So one way to quantify this is to show the relationship of the respiration to the earthworms. Now, I'm not saying the earthworms did all that breathing. The earthworms are associated with healthier soils, more microbes, more fungi, and so on. So the relationship is really important, and I think the more we can do this kind of work, and it needs more than 1-year studies, folks. It needs multiple year studies. This was a 17-year study. I happened to be a student at the research farm on the 17th year, and so we got to collect all this great data. But think of the people that managed it for 17 years in order to get the soils into these different categories of activity. So another way to quantify it, and the most difficult way, but the most classical method, is by counting microbes. And we did a lot of this in our lab over the years and have moved away from it. There's a whole issue of culturability and identification. I mean, what kind of microbe is that? See, if you're an experienced microbiologist and you know the food, the agar that you fed it, and you know the dye that was used, you might immediately deduce what the organism is, but it's not obvious. And these organisms that grow, that's a colony there. That was one cell, and then it turned into many. It's a complicated process because, as I showed you in that slide from OSU, we're talking of 1 million to 1 billion per gram. So you have to set up all these racks here, and this is like 10 to the minus 1, 10 to the minus 2, 10 to the minus 3, all the way up. So you're constantly diluting the water extract of the soil out to like 1 billion. And then you plate it, and this is these dilution sequences. And then you have to count the plates here and say, oh, that's too many, that's too many, this I can count, this is too little, and this is too few. And so you have to decide which plate to use to count, and then you multiply it by 1,000, 10,000, 100,000. That's how you count them. And it's complicated work. It's slow. And each agar may be specific only to 1 or 2 types of organisms. And there's all these soil microbes we discovered that are not culturable. We cannot grow them in the lab, which means we can't even measure them in the lab. That's why respiration is so significant. If you just use soil as is, those organisms are alive and breathing, and when you capture the CO2, you're actually measuring them, but you can't see them individually. Here's all the technology I have messed with over the years just to quantify soil microbe respiration. And if any of you have soil labs, you probably have seen most of this equipment. Beginning in the early days, it was Petri dishes, and do you know that Petri dish was invented by Mr. Petri in 1887? But anyway, that was when people started counting microbes. When they first, and they didn't even know they were involved with transforming soil nutrients until well into the 20th century. This is what's so amazing. We didn't know after we had launched the fertilizer industry. We only learned this after launching the fertilizer industry. And then they did manometers were invented in the teens, early 1900s, to measure this by gas displacement. Then we went to flux chambers. And the method that caught on was titration. This is my laboratory in the 1990s doing these it was too much work. It was so much work to measure the respiration. We went to gas chromatographs. It took too long to do it. And you spent $50,000 on an instrument and you wait 30 minutes for the result. You can't analyze a lot of soil samples. And then respirometers came along with infrared, and we have one of these. It can do 10 samples simultaneously. But these are all the ways that scientists then devise to measure and quantify soil CO2. And it's actually a lot of fun to do it. And I often ask my staff to go do that method over there and do this, and I want to see that the results are similar. They're not identical, but similar, to know that the different technologies are working. Here's one that how many of you have ever had a PLFA test done on your soil? Anybody here? I see several hands. A lot of people ask me, what the heck is PLFA? Phospholipid fatty acid. It's a mind-numbingly complex way of testing soil that fascinates analysts and is popular among analysts. You basically add a chemical to soil that destroys and explodes all the microbes. So it blows them all apart into their pieces, like dropping a bomb on an animal farm. And you run it through, you have to esterify it and run it through a gas chromatograph, and all the body parts, the lipids, come out in these lines here. There's like 40 or 50 of them that get separated out. And so you look at that and say, aha, here they all are right here on the right. And they classify them by their molecular structure. And then over the last 25 years, a consensus has developed that the molecular structure could be associated with a group of bacteria, gram-positive or negative, actinomycetes, and there's one group there that's particularly significant to indicate mycorrhiza fungi. And that's why this test is interesting. You can get closer to who it is that's doing this. But there's no real consensus on the association of all these compounds that you're testing. But it's still being used, and it's a useful test. So just last week, Dan Davidson, who is a certified crop professional out in these parts, he lives in Nebraska, finished a study with us in Ward Labs. And just to show that we're all, in the end, we're measuring the same thing, we took the Ward Labs PLFA results for 6 farms, and we ranked them. Ranking means best to worst. And then we took the Woods End CO2 Respiration Health Score and ranked that best to worst for 6 farms in Nebraska. And look at that correlation. And the beauty of this is that 2 completely different sets of procedures are indicating the same thing. He was able to rank these farms from better, based on methods that any scientist would tell you were highly dissimilar, but are both based on an attempt to measure microbes. So there's been a lot of debate about what's the right method to measure microbes. They say there's no one right method. There's a lot of ways to do it, and let's hope we correlate like this. This is very reassuring. You've all heard of the respiration test that I developed, or maybe many of you have. It was really out of a lot of frustration of doing these lab procedures that I developed this. I was the one, as a young grad student, they sent me to the cellar to titrate respiration because I was willing to do it. And later we got into some of the bigger equipment, but it was too slow, so I couldn't commercialize it. I said, heck, we should know enough about chemistry to know how to trap CO2 rapidly, which we do here in 2 different ways, and create a color scale. And the color scale was just based on a chemistry principle called the Beer-Lambert law, which is as CO2 absorbs into a chemical that has an affinity to it, it's going to change the composition of the chemistry, which can be indicated by a color pigment system. And colorimetric analysis is very common and is used widely in the soil test industry. And we created kind of an arbitrary scale for it of 1 to 5, or 0 to 5. 0 being a completely lifeless soil. I've never ever had a soil test 0, but the probe starts at 0 here at the beginning of the test. And every color unit here is a doubling of respiration. So you can see the scale goes up very rapidly. And this test now, we have learned how to quantify it in the laboratory. And so now labs can run hundreds of samples. I was in one lab the other week that ran 200 samples in one day. And that was my goal with this method, is we started at the beginning here by pointing out that soil labs are undergoing change too. And what they have told me for the last 20 years is we won't do biology methods because they're too time consuming. So you see, just like you guys innovating methods on your farm so it's not time consuming, we did the same thing in the lab to try to make this possible. And all they have to do is put it in an incubator that's warm, the same temperature, and measure it 24 hours later. So some of the exciting things that have come out of this that relate to quantification. The test on the left is 4 soils taken right up and down the road from Ray Steyer's farm. Ray Steyer's been featured in NoTill magazine, I think, at least 2 times. He was one of the very early adopters of NoTill, and in the last 10 years, his work with cover crops has been amazing. I've been on his farm twice and tested it. And this is one of our new tests for amino nitrogen in soil. And again, I designed it so the chemistry is visual, so you can just look at it and have your result, but you can also quantify it if you're a laboratory. And here's Ray Steyer's topsoil right here, right down the road from him, a truck farm, same soil, absolutely no biological activity. Heavy tillage, multiple crops a year, lots of fertilizer. Ray is almost fertilizer free, and I'll show some numbers, but it's been calculated by NRCS that has looked at his farm and his yields that he's probably picking up 105 pounds per acre of free nitrogen every year his soils are so active. And he uses it. He likes to josh with people and say, you know, I don't care about my yields, it's my profit that matters, and my profits are good because my rates are low. Ray's subsoil was even better than this guy's topsoil down the road. It started lighting up a little, which means there's this humus nitrogen. This test of mine, I mean, it's been subjected now to all kinds of university evaluations. And University of Connecticut found that by measuring this form of organic nitrogen in the soil with our tests, we can put yield and photosynthesis index into categories of low, moderate, and high. And if your soil tests in this high range, the usefulness of adding further nitrogen is questionable. They call it a soil that has a very high probability of releasing up to 150 pounds of nitrogen from biology alone. And they're telling growers, if you can get your soils up there, pull back on the nitrogen throttle. You don't need it all. We're just completing a study with 3 universities in the Northeast on dairy farms, and we're finding some of the soils are so high in biological activity, we're almost alarmed at the nitrogen mineralization. It's even more than their crops need. And this could be an issue, for water quality. Imagine blaming healthy soils for excess nitrogen mineralization. It's a fascinating syndrome, and half of the farmers are still being told to put more nitrogen on. And I'm saying, this is crazy. But they're saying, well, quantify it. You can't quantify it, you can't help us out. And that's why all this work is being done now. So this is kind of a busy slide, but so I decided, let's take it into the field, and we created this new test that you can do yourself and measure in pounds per acre per day of the CO2 coming out of your soil. And people do it, and I get lots of phone calls. They say, I can't believe it. And it was either too low, they're really upset if it's really low, or it was very high, and they wanted to know why was it very high. And we took one farm and mapped the CO2 every day through the whole season. This was a good growing season, moist soil, and look what it looked like. So the soil, as it warms up, is pushing out more and more CO2. So it's highest in the midsummer and then declines as the soil cools down. Of course, if you had no moisture in the soil, you'd have some very low numbers there. So what we're trying to do is just to move this kind of paradigm into soil testing and get people to start monitoring it. We're not saying we know exactly how to interpret it, but to start looking at the life of your soil is one of the important things. So it all comes back to this slide that I showed you earlier. Is a healthy soil capable of providing your plants with enough carbon dioxide for photosynthesis? Because if it is, it's also providing it a significant amount of nitrogen and of phosphorus through the release in the nitrogen mineralization and phosphorus mineralization cycle. And I think this is one of the most important things that we can really dwell on now in showing quantitatively the significance of this. This is not just an academic interest, is your soil alive or not? We want to make it practical so it can help you reduce some of your inputs. Going back to that critique study from 1975, they said the problem was all the nitrogen inputs. That's where most of the energy was going that weighed certain farms down in terms of the efficiency of their yields. And this is a question today that if we could do some of these energy budget studies on no-till cover crop farms and compare them to conventional neighbors, I bet we would see some dramatic differences. Proof by example, this is Ray Steyer standing in his corn. That corn received no inorganic nitrogen fertilizer. It's hard to believe. Now, I pressed him and I said, there's got to be other nitrogen inputs. Well, there is. He has a animal replacement program, and he spreads the manure on the farm, and we quantified all that, and it's only about 40 pounds of nitrogen total going on in the manure, because he doesn't spread it every year. He does it the farm gets all the manure every 4 or 5 years. So if you spread that total amount out, it could be 40 pounds a year, and he's picking up at least 100. His soils are significantly more active than neighbor soils, and we paired them with neighbors and looked at them. His CO2 respiration is 2 to 3 times any of the neighboring farm soils on conventional management. End mineralization is very positive. Nobody's done an actual study there, but we've measured the yields. Ray Archuleta used to talk a lot about Ray Steyer's farm. They were neighbors at one point virtually, and did a lot of the measurement. And the evidence of nitrogen mineralization is very, very strong. And this is how he's been able to lead to the total elimination of inorganic nitrogen. Isn't that amazing? And we're seeing that in some farms I'm studying in Pennsylvania and some of these New England dairy farms. Their nitrogen cycle and their humus is so active, they really don't need the nitrogen fertilizer unless as a starter in the spring when the soils are cold, and under some circumstances where they're trying to go for really high yield. But it's only as we begin to quantify the health cycle of the soil that people are appreciating this pressure of nitrogen and how you could actually overload your system with nitrogen if the soil is healthy. Why would you put more nitrogen on a system that already has enough? And the interesting thing on his farm, and I've been comparing his soils with some virgin soils, is the organic nitrogen pool. We call it the amino pool. It includes protein and a variety of amino compounds that are sticking onto humus in these interesting and amorphous ways. This pool has continued to increase on his farm. What that means is he's actually surpassed the native condition of the soils in some areas near him. The soil is actually better as a result of his no-till cover cropping than it was just in the hedgerows and the trees nearby where there's been no human intervention. And this is tremendously uplifting when you see this, that you can manage your farm so your land is getting better than it was in the native state. And better, not just to make you feel better, but he keeps saying it goes to the profitability of the operation. He got into no-till because he said, I was just doing too much work. There was too many implements, too much to do every day. No-till saved him time, and he said, then I started noticing the changes in the soil. And then when he brought the cover crops in, he said it really picked up. The legumes are a big part of it too, obviously cycling more nitrogen into the system. And he said, heck, no more nitrogen fertilizer, I'm fine. The fun part about this, at the end of the day, you may measure yourself with the numbers, the quantity of acres that you can get into no-till and particularly cover cropping. And I'm measuring it in the quantity of soil labs I can convert to starting to offer biology tests. And I do most of my time just traveling around what we call business to business. I meet with soil labs. They're my competitors. I don't care. We have a collegial way of relating. Obviously, we have something to sell to them, and they have a reason to want it, to give it to their customers. The customers are you, the growers. The first thing that soil labs often tell me is, nobody asks for soil biology tests. And I said, have you ever asked? They said, well, we wait to be asked. I said, well, try asking. You know, and a lot of these companies do very little marketing because they've got an audience of people that have been doing testing for decades and just continue to do it. So it's like trying to turn a ship at sea. It's slow. But these represent a lot of major labs here that have come on board with soil health testing, soil respiration testing. And we've started our own proficiency program to monitor it. So we're comparing labs statistically. How similar are they? And you can go on my website and download our latest report on 28 laboratories' performance. And it's really great. You won't see their actual names, but you'll see the range of values because there's been a lot of criticism, some of it deserved by the academic community, that we're not all monitoring these new methods well enough. We have to assure that they work as well as the old methods. So I think we're meeting that expectation and exceeding it now. And these laboratories now are coming forward and saying, this is actually going very well. One of them told me the other day it's one of their biggest growth areas in soil testing is the interest in soil biology. And they say with that comes the new methodology. This year I will introduce to the labs a new rapid aggregate stability test, and that's really important because you can see the soil structure with this simple test. I looked at the old tests. They take 2 or 3 days. Nobody's going to do them in a commercial lab, just like a farmer wouldn't do a method of tillage that hurts the system and takes too much time and money. These labs need things, and they're asking, okay, we'll take a look at it, and then we'll start offering it to growers so that over the next 10 years, I hope you all who do soil tests start seeing your reports change, and you'll be asking questions too. You'll be saying, well, why is that high and why is that low? Or what does it mean? And right now, the way that the interpretation is working, the labs are not saying very much about it because we're all in a data collection mode right now. Our test here, that's my test there over on the left. We go a little bit further than most because we redo a nutrient budget here. We show you the whole calculation and we infer how much less nitrogen you may need based on the vitality of your soil. And I get a lot of calls about it. And I've had a lot of growers call me and thank me. They said, you know, I reduced my fertilizer bill by $40,000 last year on my farm based on your report, and we didn't see a ding in yields. That's pretty impressive. And I say, well, don't thank me, thank the microbes in your soil, and congratulations that you have done such a good job growing that vitality in your soil so that you can pull back. But there is resistance and pushback in the academic soil science field in terms of how far we can read these reports to save farmers money. And at the end of the day, I say, let the farmers vote. The scientists should really take the backstage at that point and say, is it working or not, and ask the growers. And right now, it's really beginning to work, and we're seeing a tremendous upsurge in this way of measuring soil. And it's not meant to replace the old tests. Many of these tests have some of the old factors in them. There's similar extracts for phosphorus and potassium that you've been used to seeing, and then they add into that some of the biological stuff so that you're going to see a lot of familiarity with it, and then you're going to see some things that are very different. As a way to kind of summarize the meaning of soil respiration, I have to relate it to a number of other things that I think are very important, practical, meaningful, and measurable. Obviously, respiration in soil, by monitoring it, you're checking on the vitality of your soil and the activity of your microbes. And we're able to quantify it and reproducibly quantify it. And it's amazing that most agricultural soils are respiring between 30 and 200 pounds a day of CO2 in these lab tests. When it's over that, it's unusual soil, like 5% organic matter, 10% organic matter soils. When it's under that, you're more in the natural range. Litter decomposition is the primary result of active soils. Microbes that are present in soil are decomposing litter, and the more healthy and vital your soil, the faster they'll do it, the more satisfying for you. Plus, it's going to release the nutrients that were in the litter faster. This is nature's way of increasing its own vitality. The release of biological nitrogen and phosphorus is just an obvious result of respiration. I barely touched on the building of soil structure because I wanted to focus on respiration, but we've done multiple correlations of respiration tests, and there is a relationship to improved structure in soil because it's all microbial based. Soil texture has a huge role to play in aggregate structuring and aggregate stability in soil, so it's a third major factor there influencing it. And at the end of the day, and to tie this back into how I opened this presentation today about that fantastic St. Louis study back in the mid-70s, it was all about efficiency of nitrogen or inefficiency. They were citing us for inefficiency of nutrient and energy management. And this method of farming, this no-till cover cropping movement is moving us into this efficient direction, and you should be watching it and expecting to see some large growth and increases in that. So I want to end right now, and if there's time for a few questions, we will take them now. Thank you very much.
Yeah, give Will a round of applause. Will has probably got time for about 3 questions. I'm going to have you, if you want to stand over there, we're going to tear this down, but you got time for about 3 questions. And of course, you guys all know we've got lunch at 11:50 up in Archview, so feel free to take some questions. So.
Right here.
The nuts and bolts behind the Solvita test, are there months when you pull those there when the ground is warmer or not? And what are the nuts and bolts on this one?
He asked about the nuts and bolts of the soil test, whether taking samples in warmer and cooler soil makes a difference. There's different ways you can do it. One way I do it, I tell people, take samples in cold soil and measure them in cold soil, which means don't bring it into your 75-degree house, but put it in the barn and then take it in under warmer conditions and measure it at the temperature that you're taking it, and you'll begin to see those curves that I talked about. You'll see how low respiration is in the spring. But if you bring it inside and warm up the soil for just one day and run it, it's going to look like summer soil. And we have all the laboratories doing it at the same temperature, so it levels out the seasonal differences to a large extent. The soil looks the same pretty much. The difference is if you sample in summer and there's an active cover crop, the soil is so alive. When you do the respiration test, you can get very high numbers. The same soil in the fall, when all the roots are dead, you bring it into the lab and warm it up, you will not get as high of a number. And that shouldn't alarm you. That's because there's less happening in the sample. Question right here.
I thought I saw your presentation. When you get the soil sample for that test, is it the top 3 inches you take?
You can take a soil to any depth. I encourage people to explore the layers of their soil by sampling 3 to 6, 6 to 12. And you can see some sharp profiles, like in many soils, all the life is in the top 4 inches. I've seen soils that the top foot was equal in respiration because we took it in segments. So we've had students go out and sample in these depths and then line the tests up in a row, and you can just look at them and just see it declining rapidly as you go down. But one of the things you should expect, particularly from cover cropping, is the soil life is moving down into the soil over the years, and it's enriching the subsoil. And that was some work I did in my grad studies, as we showed effects down to 2 feet deep over 15-year study that were not present under conventional management systems. So the only way to do that is by taking samples at different depths. So any way you want to do it is appropriate. Just do the same thing with each sample then. Use the same method if you're comparing fields. Question here.
Just beta tests available and practical for on-farm use for us to do it ourselves?
I mean, you can buy a little package of 6 tests. Oh, the question was, is it available for on-farm use in a practical way? We have a version of it that's used in laboratories, and they have a device that reads it automatically, but it's still a good old-fashioned colorimetric test with a color chart that you can do yourself. And if you do a lot of them at side by side and lay them all out, you can photograph them and preserve it that way. And it's actually, the human eye is amazing. We can distinguish small differences in color as accurately as a photometer when you have color, a range of colors side by side. So I tell people, don't ever just do one test. Do a couple at the same time, and then lay them side by side. You can see nuances of differences there.
And where would you find that? Where you can access that on your website? Or?
Yeah, if you go to solvita.com, and we also have a store, and you can buy a home pack for it. That green kit I showed has all kinds of other things in it, sampling tools and devices, thermometer because I like people to measure the temperature when they sample, because a warm soil is way, way, way more microbially active than a cold soil, and you need to understand that. And that's why this has been challenging for the soil testing industry, because if you go to a conventional soil lab, they'll tell you it never occurred to us that temperature had anything to do with anything, and so on. Microbiology is all about temperature and moisture availability. Back here.
Do you have any comment on if you had your corn and you had a early planting of cover crop seed in there, and your cover crop is now up with a canopy underneath the corn, on that still day when you've got all the CO2 respiration coming up, is your cover crop a benefit or a harm at that point for that CO2 getting to the corn plant?
Thank you. The question was whether a cover crop would, in effect, compete for CO2. with the corn plant. The chances are not, because the biomass of the cover crop, if you did it in pounds or tons an acre, would be really small compared to the corn. And the other thing is, and my critics keep saying, come on, there's a lot of CO2 in the air and it's just going to blow in and level out the differences anyway. But potentially your soil can supply easily all the plant needs. And that would be certainly something interesting to partition out. Doing scientific studies to measure those things are very complicated because they have to put canopies in the fields and over the individual plants. But people are doing that now because this is interesting to a lot of scientists. Question right here.
Could you do this out in the field with the jar, turn the jar over and put the tablet in it?
Jill Clapperton likes to do it that way. If any of you have seen, she takes our test and just puts the jar upside down. And what it does is the CO2 is rising up out of the soil, diffusing out of the soil, and it gets temporarily trapped in the jar. And you'll get a color change, and it's a meaningful color change. The problem with that is it's not a sealed system, right? So that as it builds up in the jar, it reaches a concentration higher than it was in the soil. Then it starts diffusing back the other way. So we find it tends to level out. But I've seen people doing it in the field and doing it like in the shade and in the sun. And you can see huge differences right there. And that can be meaningful because, I mean, the biology of the soil is being influenced even by patterns of where sunlight is falling. So these heavy canopied soils are actually cooler soils, and they're releasing less CO2, which is very interesting. I guess we're all set, and it's time for lunch. Thank you all again very much.